morning, everyone. Yeah, I'm going to be talking a bit about the Woodland Myotis project, which was a project between the Glamorgan Bridgend and Cardiff bat groups that we set up back in 2019. We had quite a hectic year that year getting this project going and getting out surveying lots of sites. Unfortunately, because of the pandemic, we haven't done much in the way of surveying for it this year. It's all been cancelled by Corona, like everything else in our lives. So yeah, just going to be talking about what we did that year and what we're hoping to do when we can get stuff started again. So just for anyone, I'm sure people are quite familiar with bats, but for anyone who doesn't know, we've got 18 species of bats in the UK and they fall into 7 genera. And most of those different genus of bats have only 1, 2, maybe 3 species, but our Myotis genus has 7 species in it. So 7 out of our 18 UK bat species belong to this Myotis genus. So they make up the bulk of the biodiversity of bats really in the UK, with being about 40% of our UK bat species being Myotis bats. So what is a Myotis bat? How do you know you're looking at a Myotis bat? So if you've got one in the hand, it's a lot easier. They are what we call like a plain-faced bat, as in they don't have any nose leaves like a horseshoe bat has. So sort of a plain muzzle. You can see in these pictures in the right-hand corner. They have a long pointy tragus, which you can also see. I'm not sure if you can see my cursor on the screen, but tragus is just a bit inside the ear. It's easier to see on the bat than it is on me. And that tends to be very long, pointy, triangular shape in Myotis bats, whereas it tends to be blunt and a bit shorter or maybe mushroom shaped in other bat species in the UK. And all of our UK bats, Myotis bat species, do not have this thing called a postcalcareal lobe. So you can see on this diagram here, this is the leg or the foot, and the calcar is basically like this bony filament that comes out of the foot and extends partway along the base of the membrane that links the tail and the foot or leg. And some bat species have something called a postcalcareal lobe, which is basically just a flap of membrane under that calcar. But our Myotis bats don't have this. So if you've got a bat that you've found and it's in your hand and you can see it's got these features or doesn't have some of these features, then you're probably looking at a Myotis bat. So in the UK, the 7 species I've listed there, Daubenton's natures. We have something called a greater mouse-eared bat, but that's I think only known from 1 individual now in southern England, so virtually extinct sadly. And then we have Bechstein's bat, which is another very rare one, not quite as rare as the greater mouse-eared, but still very rare. And then we have the whiskered Branson alpha-bowie bat, and those last 3 are something that I might call small Myotis or WABs throughout the presentation. And those basically are 3 very small Myotis bats. 2 of them are in this picture here, so it's a Brant's next to a whiskered bat. And they're very difficult to tell apart even when you've got them in the hand. And the alpha-bowie bat we actually only realised was here in the UK in 2010. So yeah, quite recent finding really that new species is, or probably not that new, probably was here all along, but we've only just realised it's here. And in 2019, an alpha-bowie bat was confirmed at a swarming site around about that red pin that you can see on that map there, so kind of close to Bath, which isn't a million miles away from us in Cardiff, Vale and Bridgend counties and South East Wales generally. But as far as I know, I don't think there's any confirmed alpha-bowie in Wales yet, but they could possibly be here. So one of the big things about Myotis as well is that they're really difficult to tell apart or identify to species level by acoustic methods. So you might have gone out and done bat surveys in the past with a bat detector where you can hear the bat's ultrasound, and you can maybe look on a computer later at a sonogram. So with lots of our bat species in the UK, you can tell them apart when you're looking at that sonogram, or maybe when you can hear them with the bat detector and you can see the frequency. But the Myotis bats are really difficult to do this with accurately. So that makes it a bit more difficult for us to survey them. So back in 2018, the group was sort of looking at starting a autumn swarming project. So for anyone who doesn't know, swarming sites are, or autumn swarming sites specifically, are these sites where large numbers of bats usually congregate, males and females, in sort of late August to October time. It's thought that they're really important mating sites and that they basically facilitate a lot of gene flow. So bats, particularly Myotis bats, but also things like brown long-eared and some of our rarer bat species like Barbastells, will come from vast distances to these sites and we think mate and then also usually hibernate there as well. So just as an example of some of the crazy distances that they'll travel, there was this one study by Rivers that found that Natter's bats were coming from a distance of over 60 kilometres from their nursery roost to the swarming site. So they're really important conservation sites. There's also some evidence from some research to suggest that bats don't really like to change swarming sites, so they'll go back to the same swarming site throughout their lives. So their loss obviously would be quite serious for bat conservation efforts if one was to be lost. So back then we had no known sites in the Vale of Glamorgan, Bridgend or Cardiff. So we wanted to have a look and see if we could find any. And the way that we went about this at the time was to basically look at maps, look at online resources like caving websites, and just try and mark out any possible potential sites. So they do tend to be underground sites, things like caves, possibly like old mines, things like that, but they can also be overground features, things like old railway tunnels. So we basically just marked out everything that we thought could possibly be a swarming site, and then our plan was to systematically try and go through them, visit them during the daytime, see if they actually looked good for swarming there in the daytime, and then if they did, leave static detectors out. So that's like a small bat detector that we can leave out for a long time and see what the activity levels were like in the night, see if we could pick up swarming behaviour on those. But obviously that's crazy labour intensive to try and do it that way. That was a lot of different places to visit. And we kind of realised that we might spend most of the rest of our lives looking for those if we carried on doing it in that very labour-intensive way. But at the same time, we were also doing these monthly tree surveys at a site that we have in Bridgend, where we were doing monitoring of tree roosts for something called the Battery Habitat Key Database. And when we were there was one survey event where we found the first Natter's tree roost record. And there was about 5 Natter's bats in there, which was really exciting. And I had what I thought at the time was a light bulb moment that maybe there was an easier way to sort of nail down possible swarming sites using roost information. So as I've just said there, there's that one study about the 60 kilometres distance travelled from nursery roost to swarming sites in Yorkshire. But there's also lots of other data suggesting various distances for different Myotis species in particular. So the idea was that we could possibly use Myotis roost data to try and limit our search efforts down. So we went to Subrek and asked them if we could have all the Myotis records for our counties, who kindly obliged and gave us all the information that they had. Which was actually only 433 records, which kind of sounds a lot at first, but when you think that that's for 3 counties and up to 7 species of bats, probably more likely 6, not really very much data at all. And if you have a look at this pie chart on the right, so 40% of it was unknown Myotis species. So as I said before, because they're really difficult to identify using sonograms and acoustic methods, that means a lot of Myotis records get put into the Biological Records Centre as a Myotis species. So we don't know what species, but we know it was a Myotis. So that makes up quite a big bulk of the data. And then Dorbenton's bat made up 45% of those records. And the reason for that being a lot of those records came from something called a Waterways Survey, which is a survey that the Bat Conservation Trust run. It's part of their national bat monitoring programme, where volunteers go out sort of on a yearly basis and survey different waterways for Dorbentons. And the great thing about Dorbentons is that they forage in a very characteristic way over the water surface, so you can go out and do these surveys with a bat detector and you can hear a series of dry clicks. So you can be quite confident you're looking at a Daubenton's because of the way that they're foraging, and then being able to hear this quite distinct sound. So Daubenton's are kind of good in that way. So there's a lot of information about Daubenton's on waterways in our counties. And then that leaves us with not very many records at all. So 38 nattered bat records, 27 whiskered bat records, 6 whiskered/France records, because the smaller ones, small Myotis, are really difficult to tell apart, so they also sometimes go into whiskered/France. And then a single France record from Cardiff, which was actually from a sound acoustic file, so dubious as to how accurate that may or may not have been. And just as an example of why my light bulb moment wasn't a light bulb moment at all, so this is 60 kilometres from Cardiff, so that wouldn't have narrowed it down very much at all. And even a lot of the other research outside of natterers bats, it's things like 20 kilometres, 25 kilometres, so it really wouldn't have helped that project whatsoever, really, even if we'd have had loads of roost data. But one thing that we realised from doing that data search was that we have really very little information about Myotis bats in our counties, a real lack of species specific records, and actually of those 433 records, only 56 of them were from hand identifications. So as in someone's got a bat actually in their hand and can look at the features on it to tell its ID. So arguably some of those might not be that accurate that we have in there at the moment. We had no records for Bechstein's bat, no records for Alcatho bats. No, not that I'd expect there to be greater mouse-eared bat, but there's no records for that either. And then we had this one record for Brants in Cardiff County that was from an acoustic recording, and no records of Brant's bat whatsoever in the Vale of Glamorgan or Bridgend. And then really just a small handful of whiskered and natterers bats records. So what does that mean? Does that mean that all these species are really rare? Are they actually absent? Are people just not looking for them in the right way? And one of the other things that we realised from the DEF study is that huge amounts of that data that has been put into the Records Centre is largely, aside from the Daubenton stuff for the waterways surveys, a lot of it's coming from commercial bat surveys. So a lot of it is surveys of built structures. There's not very much survey work that's gone on by the looks of it actually inside woodlands. And woodlands are important habitats for bats generally, but they're particularly important for all our Myotis species. So we thought we need to go and have a look for some of our Myotis bats in some of our woodlands. So the project aims were to basically determine whether some of these species are present or not in our counties, things like Brant's, Alcatho and Bechstein's bat, and how far species distributions in our counties. You can see here this table that I pulled from this Ray et al. Paper from 2010. Generally, it looks like, based on that data at the time, that a lot of the Myotis species are rarer in Wales. You might have seen recently the Mammal Society brought out a State of Mammals in Wales report. A lot of the Myotis bats and a lot of the bats in general actually sadly were just data deficient. So that document tries to look at what range trends are happening with different mammal species in Wales, population estimates, population trends, and a lot of them for the bats were just data deficient. So it's just not enough data about a lot of our bat species. And we're also hoping to identify important sites, so whether some of our woodlands are really important for certain species of Myotis or not. And because in our counties we benefit from having a continuous strip of coastline, we also wanted to have a look at whether there's other ecological factors that determine species presence. So things like coastal versus inland woodlands. Do we find differences in bat species assemblages, whether it's a more coastal woodland or whether it's more inland? And the same with things like woodland types, so whether what kind of bats we're going to find in plantations, broadleaf, ancient, etc. And then woodland use. So because we have to use trapping in this, I'll get onto the methodology in a second, but we did use trapping for this project. It also gives us the opportunity to look at other aspects of bats that we don't get to record with a normal traditional bat detector survey. So that's things like whether they're male or female bats, what kind of reproductive state they're in, whether they're pregnant, breeding, lactating, etc. So we wanted to have a look at all those things. So yeah, methodology-wise, because we can't use bat detectors for this, it's not going to give us the species-specific information that we're after. We went to trap bats in woodlands using heart traps, mist nets, and acoustic lures on the heart traps. So if you're not familiar with these, this top picture here, that's 3 bats that have been caught in a heart trap. And you can see the heart trap set up in that bottom right picture, and then quite hard to see the mist net, but mist nets are the same kind of mist net that you use for bird mist netting, if you're familiar with those. And yeah, we started with 9 sites, and the idea was that we were going to try and do 3 surveys per site in each of these different bat seasons, being the pre-maternity period. Then we have a little break where the bats are really heavily pregnant, they're having their babies and they're small and very dependent on them. So then we have a little break to give them a rest because we don't want to stress bats out in that state. And then we start trapping again in the post-maternity period, and then once again in the autumn period. So at the end of 2019, we'd managed to get around 5 different sites, not all in their entirety, but we managed to get around 5 different sites, and we managed to get 20 volunteers out training with us, doing some bat handling and some trapping. We caught 123 bats in total, and of those we had 9 species of bats, and of the 129, only 29 of them were Myotis. So the only annoying thing about the trapping surveys is that we can't specifically trap just Myotis bats, so we get a lot of bycatch, so that's why we got 123 bats, but only 29 of them were Myotis. And we got 4 different Myotis species, which were whiskered, brants, natterers, and Daubenton's. And one of our most exciting findings was we got our first proper record for a brants bat in Bridgend County. And that bat we had, so we collect droppings off of the small Myotis bats so that we can send them off for DNA analysis. So we had that confirmed with the DNA analysis, which is what we're looking to do wherever we can, really, is get a dropping for any of those small Myotis bats, because they are difficult to tell even when you've got them in the hand which one they are. And we also had a site where we had a lot of pregnant whiskered bats, which was quite exciting because I suspect that there's probably a whiskered bat maternity roost somewhere in there, and I don't think we have any record. Oh no, I think we do have 1 record, but we don't have very many records of whiskered bat maternity roosts in our counties. And we also had, one of our woodlands had quite high levels of Myotis activity in the autumn, which was quite interesting when you relate it back to our previous swarming stuff. So the fact that there's still Myotis hanging about in fairly large numbers in the autumn kind of suggested that we're either close to or sort of within the vicinity possibly of a swarming site. So obviously this project just started and now we've had to put it on pause a bit. So we don't really have enough data to do any sort of fancy statistical analysis yet at all. But just as an example of some of the things that we're going to be looking at, so site A was a coastal site, broadleaf woodland, and it also had some areas of ancient woodland in it. Site B was an inland site, also broadleaf woodland. It was actually a country park, and it had some areas of wet woodland, and we didn't trap on the river, but there is a river that goes through site B. So just we can start looking and comparing our different woodlands basically in our counties. So at site A and site B, you can see that we had similar numbers of species. You can see soprano pipistrelles made up the bulk of the catch. And we had 2 Myotis species at each woodland, but 2 different ones. So we had whiskered bats at both sites, and actually we got whiskered bats at most of the sites we trapped at, so they seem to be definitely more widely distributed. And we had Daubenton's at site A, the coastal site, and we had natterers bats at site B, the inland site. And we can also have a look at things like sex differences. So there's actually quite a lot of literature that's worth having a read into if you're interested on sexual differences between sort of ecological niche partitioning, so males and females using habitats differently. So And if we've got lots of females, that can be an indication that we're looking at higher quality habitats or the most productive habitats for that species, because the females tend to dominate these habitats, particularly when they're pregnant or lactating, when they've got higher energy needs. So at site A, we had more males than females. And at site B, we had more females than males. So again, that's kind of another indication that site B could be a very productive habitat. And that site is also the site that we had a lot of pregnant whiskered bats at. And then the same can be done with reproductive status, so basically taking it a stage further. This is kind of a bit misleading, these charts, because I've pulled the 3 different bat survey data all together, so it looks like there's an awful lot of not breeding animals, but that's because the surveys later on in the year, in the autumn, had obviously a lot of juveniles flying around who weren't necessarily in breeding condition yet. So yeah, you can see site B, we had quite a lot of pregnant animals, or noticeably pregnant animals, and site A, we had quite a few post-lactating animals, but not any pregnant animals actually. And then we can also do things like look at how these different factors change throughout the year. So as I said, that last one was a bit misleading, so I pulled them. So you can look at different aspects throughout the year, which is something that we're hoping to do. So just taking site B, which was the inland site, as an example, you can see here that the species composition has changed quite wildly over that summer. So in the early season, we had only 3 species, and only 1 being whiskered bat's Myotis species present. And then in the middle of the summer, we see it jump up to 6 different species of bat. And we've also then got Natura's bat, another Myotis bat there in the middle of the summer. And then in the late summer, it drops right back down again. But I think we still had, yeah, whiskered bats still present. So whiskered bats present throughout the whole year, it seems, or the whole summer rather, at this site. So we had big plans for 2020, which obviously haven't happened. But we were hoping to really expand, continue surveying sites, gathering more data. We had new sites that we wanted to add to the project. So in 2019, the same year, we also started taking part in the Welsh Enthusiast pipistrelle project, which you might have heard of, where we were trapping water bodies for Enthusiast pipistrelles. And during those, we actually caught what we thought was a female brant. Unfortunately, she didn't give us a dropping, and we couldn't confirm it with DNA. But we are really keen to add that site, to go back. There's lots of woodland there, so to go back and trap in the woodlands and see what we get, and see if we can confirm brants in the Vale of Glamorgan as well. And into the future, we'd really like to radio track some of these rarer species, things like the brant's bat, or seemingly rarer species. Because we don't have any records of them, full stop. We don't have any roost records for them either. So that would be really interesting. And we'd also like to expand into other counties in southeast Wales, particularly if they've got similar data deficiencies for some of their Myotis bats. And then for some of them, site-specific projects have come out of some woodlands. So we're working with woodland owners and managers to manage their woodlands and make them better for bats and protect any bat interests that are already there. We're also hoping to do some more biological recording, to do some moth trapping. So the plan for 2020 was to try and invite moth trappers out with us, because I think that would just be a really interesting extra element to the study, looking at moth biodiversity and abundance at these sites when we're also trapping for bats, and see how that correlates. But also because it's just an opportunity, we've got access to all these sites, and they're really wide ranging. It's everything from country parks, private woodlands, triple SIs. It just makes sense to also be gathering more data for other under-recorded groups whilst we're out there. And then lastly, one of the big things that we're interested in Myotis in general, but the small Myotis, those whiskered brants and Alcathoe that are hard to tell apart, particularly interested in trying to understand if they're present, where they're present in our counties. So we've also created this small Myotis DNA analysis project, where we've been really fortunate to receive some funding from Subrec for that, so that we can carry on doing DNA analysis of any droppings, not just through this project, but through all our other bat group activities that would normally run in a normal year, things like bat box checks, roost visits, stuff like that. So if people, and also for bat carers as well. So the bat carers provide quite a lot of useful species specific records because they actually see these things up close. So the idea being that anybody in our bat groups who takes part in any of those activities, if they come across small Myotis bats, there's now a pot of money that we can send off and get those checked by DNA analysis. So that's really good. And then finally back to the swarming. So it sounds like that swarming project just got completely pushed to the side and forgotten about. But it wasn't completely forgotten about. In basically one of the things that we realised is that this idea of trying to narrow down our search and make it a bit less labour intensive was not going to work. So really what we needed was more manpower and more equipment to go out and look at these sites. So we were really lucky at the beginning of 2020, before the pandemic had kicked off. We won some funding from the Bat Conservation Trust for some audio moth bat detectors, which are these teeny tiny bat detectors that you can leave out on sites for whatever length of time, and then go collect, and it's recorded lots of bat activity. And then you can have a look at the recordings and see what you've had flying past. So with swarming sites, there tends to be a peak of activity 3 to 4 hours after sunset. So the idea being we go and deploy these bat detectors, and then 3 to 4 hours after sunset, if we're seeing peaks of activity, that's an indicator that we might have a swarming site. So the idea was we were going to run workshops and get people involved going and deploying these, doing site investigations and then deploying detectors at any good sites. Then the pandemic happened and lockdowns happening here, there and everywhere, and it just basically didn't really become feasible for us to do it as a group event. One thing we did manage to do was I managed to get out with a couple of the detectors to a site locally. I think that was in August or September, and managed to leave them out for a few weeks. Just by absolute chance and luck, this site seems to actually be a swarming site. So we found significant Myotis activity on consecutive nights with those characteristic peaks of activity 3 to 4 hours after sunset. Oh no, I've put September, so I must have done it in September apparently. So yeah, so that's really exciting and really, really lucky, because that was the only way we managed to really get out doing any part of that project this year. So yeah, massive thank you to everybody who's helped out with this project. There's been lots of different people who have helped fund it or provided data, and volunteers who have come out with us, and obviously all the site owners and managers and everyone in the back groups. If you want to get involved, we're not really doing that much right now for this project, but you can email me and I can add you to the mailing list, and hopefully it won't be too long before we're doing stuff for this again. Or you can also check us out. Cardiff Bat Group's got a Facebook and a web page, and so does the Gwent-Glamorgan and Bridgend. So you can have a look on those and join the Facebook pages to keep up to date with other stuff that the Bat Groups are doing at the moment or trying to do at the moment. If you're interested, those are some really good papers that touch on in more detail some of the things I've sort of briefly whizzed over to do with swarming and Myotis bats and sexual ecological differences between bats and things like that. So if you are interested in reading any more, there are those. And yeah, hopefully there might be time, yeah, for 10 minutes questions.
Brilliant, Jess, that was fantastic. Not a subject I'm massively familiar with myself. Obviously going into quite a lot of depth on the Myotis bats, and as I was told in the back in the summer when I was using one of Subrek's lockdown purchases, which was one of those little Echo Meter Pro bat detectors, and I was getting Myotis coming up, and of course as soon as I spoke to a bat expert, they said don't just record it as Myotis spoof for the time being. So that's the general advice, isn't it, if you're holding a bat recorder, bat detector. I've got a couple of questions from the Q&A, which I think I'll ask myself rather than try and make people go live and be presenters. So the first question was from Peter Sturgis, and it was, is a Myotis bat too small to carry a GPS tracker to follow to swarming sites in autumn?
Oh yeah, so GPS, they are too small to hold those, unfortunately. So the small Myotis bats, just to try and give you an indication, are sort of only like that from head to, well, bum really, because then there is the tail. And even the bigger ones, like our Natchez bats and Dorbenton's bats, they are a bit bigger, but they're sort of like that. So they're really tiny, really, sort of looking at like 10 grams, like, yeah, it's just not big enough. But radio tracking we do of bats, so that is an option.
Okay, so you can do radio tracking, not GPS tracking. Okay, great. The other question, I think you probably answered yourself. It was from Mark Butler, and he said, "Are there good ways to get involved in this project?" And I know you said there's not a great deal going on at the moment, and you gave out your email address there, which I've put in the Q&A, and I think other people might be able to see if they click on the Q&A. So that will be there afterwards. So is there anything else they can do at the moment other than email you?
Yeah, I mean, checking out the Facebook pages is probably the best way to keep up to date with what's going on. It's really unfortunate at the moment. Basically, the IUCN specialist group for bats have said that unless it's extremely urgent, no research that involves direct handling of bats can take place, because they're concerned about us transferring COVID to our native bats and then it having possible consequences. So everything that we did that involved direct handling is currently on pause, except for bat care, because that's considered essential. So yeah, but we are hoping this year that we are going to be running some projects, but they probably just won't be group projects where we meet up with people in the same way. So we're hoping to do a number of acoustic studies that we're trying to seek funding for at the moment, which people will hopefully be able to get involved with, looking at Barbastell and also bat migration around the coast. And we're also with Cardiff Bat Group doing an online education programme that if people are interested, like just delivering like short half-an-hour virtual talks for schools and children's clubs. So if anyone wants to get involved with anything like that, if you have a look on our Facebook pages, there's some information.
That's great. Perhaps you could paste a link to your Facebook group in the chat once you finish your presentation. That would be fantastic. Another question's come up on the chat. That's from Stephen Davison. Question: did you detect Barbastell with bat detectors?
No, not yet. I think in the Vale of Glamorgan and Bridgend, we don't have any records of Barbastell. I know that there was one where when we did a desk study with Subrek, but this was probably a year or so ago now, so there might be more now. But there was one record logged with Subrek for Barbastell in Cardiff, but I know that there are other records from consultancies that haven't submitted them to Subrek, unfortunately, yet. But yeah, so there definitely is some records in Cardiff, but the other counties not so sure.
Okay, that does flag up the issue of something which we're quite often mentioning, especially for those involved in consultancy work, the importance of feeding back records as and when they can. And obviously there's sometimes permission issues over sharing data that's been captured during consultancy work, but it's absolutely vitally important that it makes its way into the system so that it can inform other future work. I just wanted to say there are people saying hello in the chat from all over South Wales, Cardiff, some people from over the boundary in England, and we've even got somebody listening very intently from, I think it was from Beirut. It was a bat specialist at university, I think it was old Palermo. So brilliant, lots of lovely positive comments in the chat, which you'll be able to read back later. Does anybody want to ask one quick last question?
There's 7 more questions in the Q&A panel.
Q&A, okay, I've not refreshed that. Okay, I've done that now. So I'm going to have to pick and choose those. We've answered Stephen's on there. So I'm going to pick Rob Thomas's question. Rob Thomas from Cardiff University. Any advice on how to collect droppings from bat boxes without opening them? And where do you send them for DNA analysis?
Without opening them is a bit tricky. Probably depends a bit on the style of the bat box. If you've got sort of like an open-ended one, I guess you could try and maybe put something lower down that might catch them. That would be my only thing I could think, is if you've had nothing that obviously blocks a bat box, but maybe like half a metre down or something, if you could try and install something. Or you could, if you need to open a bat box, unless there's maybe a design that you can't open, you could also, I'm sure the bat groups would be happy to help out if it's just a case of needing somebody licensed to open them. But yeah, it's a bit tricky. It's a bit tricky with bat boxes as well, because the droppings can wash away. So if you have got an open bottomed one and you've got droppings falling out even onto a ledge, you probably have to check them quite frequently, otherwise they might get eaten by insects and stuff. In terms of who, I'm trying to think. Oh, Swift Ecology, that was it. Swift Ecology is where we send. I think there's a lot of different options now for sending droppings off for DNA analysis, but we've used Swift for this project and they've been really good.
Fantastic.
Yeah, we have a discount, I think, because it's a bat group. But I think if you're not necessarily commercial, they might be able to offer you a discount as well. So yeah,
great, thanks, Jess. Two similar questions. Alison Jones in the Valleys Bat Group area and Mark Butler, who's over in Monmouth area, asking, Alison's saying that the Valleys Bat Group would be interested if you wanted to look at sites in the Valleys area, and there are lots of potential swarming sites there. I guess you're probably just focusing on your own area for the time being, but obviously other bat groups in the area would be interested in what you're doing. And Mark, yes, asking if you'd be interested in expanding the project in the Monmouth area. So I think it's just, they're just generally positive comments. I don't know if you've got anything to say in response to that. I guess there's only one of you and or a few of you, and limited.
It would be really great if people are interested, when we're allowed to handle bats again. It would be great to do some kind of event, maybe in the valleys, training people up in some of those methods. If people are interested, that's what we would like to do. So yeah, that would be grand.
Fantastic. Stephanie Coates asks, what is the point of swarming?
So, well, what they think it is, and there is some quite good evidence actually to back this up now, is that it's basically a mass gene flow. So it stops bat populations inbreeding. So all these bats come from all over the place to these single sites, mate together so that they can mate with lots of different individuals, and then they can go about the rest of their year back from the places that they came from. So yeah, I think the real thing is that it's probably gene flow, but I mean, they're also, yeah, they tend to also hibernate at those sites as well. So they're kind of important for a few different elements of the bat life cycle.
Fantastic. I was going to shout out one last one. Kelly Thomas says that whilst there are limitations, is there a way to work with automatic passive acoustic monitoring, e.g. audio moth, to get more records for sites and get some long-term monitoring? She says I've been chatting to BCT and they're intending to have an app for this soon. That sounds quite technical. If you think you can give a non-technical answer, or perhaps you could type a more technical answer to Kelly afterwards.
Yeah, I mean, I think there's definitely value in, I know it kind of, from what I've just said, I'm like, oh yeah, trapping's so superior, but I don't really mean that. There's definitely a lot can be gained from acoustic methods. It's just, the only problem is with those Myotis bats specifically. And even, to be honest, sometimes you can actually work out a Myotis species from, if they've made a social call as they were passing. Those ones tend to be a bit more specific to the species, so it's easier to narrow down. So I think there's definitely value in it, and different things that you can look at, sort of like activity levels of Myotis or other species specific things. So I think, yeah, there definitely is useful. It's definitely worth doing acoustic passive stuff.
Fantastic. Thank you very, very much, Jess. That was a brilliant presentation and obviously sparked a lot of interest